Dr. Donald Yates had spent years building genealogical databases. He wasn't looking for controversy. He was looking for Cherokee matrilines, genetic lineages passed exclusively mother to daughter, unchanged for thousands of years, untouched by the 1838 bottleneck because they ran through unbroken female descent, going back further than any government march could reach. He assembled 52 participants, all documented, all with matrilineal Cherokee descent they could trace through their mothers, their grandmothers, their great-grandmothers. He sequenced their mitochondrial DNA. He expected what every genetics textbook since 1993 said he would find: haplogroups A, B, C, and D. The four markers that, according to mainstream science, account for all pre-Columbian Native American matrilineal ancestry. He did not get them. Now, those four haplogroups showed up in less than 8% of his sample. The other 92%, something else entirely. The most common unexpected result was something called Haplogroup T. Haplogroup T has a nickname. Geneticists call it Terra. It was named by Oxford geneticist Brian Sykes. And Terra didn't come from Northeast Asia. Terra originated in Mesopotamia, roughly 10,000 to 12,000 years ago. Today, its highest concentrations are in Egypt, Israel, and the Levantine coast. It spread north through the Caucasus Mountains, west into Mediterranean Europe. In every standard model of human migration, it has no route to the mountains of North Carolina. And yet, there it was, in nearly 27% of Yates's Cherokee sample. But Yates didn't just identify the haplogroup. He checked the specific haplotypes, the precise genetic fingerprints within Haplogroup T, against every database available. The Cherokee versions don't match modern Europeans. They don't match modern Middle Easterners. They don't match anyone currently living anywhere on Earth. They form a branch that split off from the main T lineage before the historical period, before Rome, before the Old Kingdom of Egypt. When Yates expanded to Phase 2, 67 more participants, Haplogroup T stayed at 22% of the sample, and then something else appeared, Sub-Saharan African Haplogroup L, at 9% of the total, with mutation levels too deep to be explained by colonial-era admixture. Before Egypt raised its first pyramid, something was already moving, and the Cherokee genome may carry the record of where it went. But pyramids are the wrong image. The people we're looking for built nothing that survived. Jericho, 8,000 BCE. The oldest continuously inhabited city on Earth. Haplogroup T is already present in the Levant. These are Neolithic farmers, traders, builders. They're expanding, north into the Caucasus, west into Anatolia. And then there is a third direction on the map that no mainstream textbook follows, east, toward the steppe, toward the Pacific. Here's where it gets more complicated, because Haplogroup T isn't the only anomalous marker in the Cherokee. There's also Haplogroup X, specifically a variant called X2A. Dr. Antonio Tironi at the University of Pavia in Italy spent years mapping where Haplogroup X actually lives in the world. What he found, published in the American Journal of Human Genetics in 2003, was a pattern that doesn't make sense under the standard model. Haplogroup X2A is present in pre-contact North American populations. It's confirmed in ancient remains, but it's nearly absent from Siberia, from East Asia, from anywhere along the route that a Bering Strait crossing would have left it. Think about that. If your ancestors walked across a land bridge from Northeast Asia into North America, they would have left Haplogroup X scattered across that path. There should be X2A all through Siberia. There isn't. This finding was published in a major journal in 2003. Nobody put it next to the Cherokee data. Not officially, not with funding, not with a follow-up study. In 1996, a construction crew in Kennewick, Washington, uncovered a 9,000-year-old skeleton. His skull didn't match what scientists expected of a Northeast Asian-descended individual. The Army Corps of Engineers seized the remains. 9 years of legal battles followed. When his full genome was finally published in Nature in 2015, it confirmed he was Native American, and his mitochondrial DNA carried X2A, the oldest X2A lineage ever found in the Americas, in a skeleton from Washington State, 9,000 years old. He was reburied in 2017. The geographic gap in X2A distribution was never resolved. Whatever people carried Haplogroup T and X into the Americas left no buildings, no inscriptions, no layer in the archaeological record that anyone has named. They arrived, they mixed, they became invisible inside the populations that would eventually become the Cherokee. In every database on Earth, they are anonymous. And the question they leave behind is this: what were they running from? The catastrophe has a name, it has a date, and it is the largest forced migration in human prehistory. 8,200 years ago, a glacial megaflood, Lake Agassiz, covering more area than modern France, collapsed into the North Atlantic. The freshwater surge disrupted ocean circulation. Global temperatures dropped 3 to 5 degrees Celsius within a decade. Crop systems failed. Archaeological layers in Levantine settlements show sharp cultural rupture at precisely this moment. The Neolithic villages that had stood for centuries went dark. Researchers documented this across Eurasia, finding what they described as demographic collapse synchronized with the event. People didn't adapt in time. They moved, or they died. The Levantine populations, the people carrying Haplogroup T at high frequency, farming in Jericho, trading across the eastern Mediterranean, they were living directly in the impact zone. Some moved north, some moved west into Europe. One branch, carrying the specific T haplotype variants that match what Yates found in the Cherokee, disappears from the Eastern Hemisphere record entirely. Everything I just told you about that Levantine population, hold on to it, because what comes next is the moment it starts to mean something completely different. In 1900, a Smithsonian anthropologist named James Mooney was sent to document Cherokee oral traditions. He recorded the Ani-Kutani, a priestly caste the Cherokee described as people who came from a distant land, who held religious power, who were eventually massacred by the Cherokee for abusing that power. Mooney categorized it as mythology. He had no genetic data to reread it with. The Cherokee green corn ceremony, the Selu tradition, encodes near starvation, followed by communal rebirth as its central ritual logic. Every ethnographer who has documented it describes it as a cultural memory of survival after total loss. It is performed today. It was performed last year in Cherokee, North Carolina. Nobody has formally asked what catastrophe it might be remembering. Actually, real quick, if you watched our last video on the ancient DNA of the Dead Sea Scrolls, you already saw the first piece of this puzzle. We traced Levantine populations into the genetic record of one of the most contested documents in religious history. Now we're asking whether the same dispersal reached further than anyone thought. What did you think of that video? Did it change how you see this region's role in ancient history? Tell me in the comments, then stay with me, because the answer to that question is exactly where this story is going. The timing aligns. A Levantine branch that disappears from the Eastern record at 8,200 years ago. A population in the Appalachians that carries its genetic signature. The distance is extreme, the window is narrow, but the window is open, alive. That word is what the oral tradition has always been, a living record, and we have been reading it wrong. The Cherokee language itself is a problem for the standard model. Cherokee is an Iroquoian language, linguistically connected to languages spoken around the Great Lakes region. With a divergent state, linguists estimate at 3,000 to 4,000 years ago. By their own linguistic DNA, the Cherokee are not the original inhabitants of the Appalachians. They migrated there from the north and arrived relatively recently in geological terms. That's a completely separate migration from the Haplogroup T and L anomalies. The Iroquoian language belongs to the newer layer. The anomalous haplogroups belong to something older, something that was already there when the Cherokee arrived. There are two things this tells us. The second one is the one that matters. The Cherokee may not be a single origin people. They may be a convergence, a merger between a Great Lakes Iroquoian group moving south and a much older anomalous population already living in the Appalachian highlands. The standard genetic models have been testing the merger and describing only half of it. And here's what Mooney noticed in 1900, without any genetic context to understand it. The Cherokee clan structure, 7 clans, strictly matrilineal, has no parallel in Iroquoian culture. But this kind of clan structure does appear in ancient Levantine tribal organization systems. Mooney wrote it down. He filed it as an anomaly. That was 125 years ago. No formal comparative study has been done since. Then there's the Phase 3 Cherokee study, the largest matrilineal Cherokee sample ever assembled, closed in 2018. The preliminary results have been described publicly. The full dataset has never been published in a peer-reviewed journal. No major genetics institution, not Harvard, not Max Planck, not the ancient DNA database, has formally requested access to it. Not one. Every dataset has a gatekeeper, and in the case of Cherokee Genetics, the gatekeeper has a name. Harvard Medical School, 2012. Dr. David Reich, one of the most respected ancient DNA researchers in the world, publishes a landmark study in Nature. His conclusion is clear. All Native Americans descend from a single founding population that crossed the Bering Strait approximately 15,000 years ago. Any anomalous haplogroups found in smaller studies are almost certainly the result of post-Columbian admixture, Middle Eastern and European genes that entered native populations after 1492 through intermarriage. Case closed. Reich's methodology is the global standard. His sample sizes are large. When someone at Harvard says the anomalies are explained, the field listens. Funding for independent Cherokee matrilineal research effectively stops. The Yates work is classified as fringe. The debate is filed away. But here's where it gets strange. Dr. Jennifer Raff at the University of Kansas published a best-selling book in 2022 called Origin, A Genetic History of the Americas. It is rigorous and well-sourced. Haplogroup T in the Cherokee is not mentioned. The DNA consultant studies are not cited. Alternative theories are described as myths in the introduction and not revisited. The omission is not a typo. It is a position. In 2017, the Eastern Band of Cherokee Indians issued an official statement. Commercial DNA tests cannot determine Cherokee citizenship. The tribe does not recognize DNA as a basis for enrollment. One, this protects real people from a real industry that has consistently exploited indigenous identity. And for a researcher trying to access pre-contact Cherokee skeletal samples for ancient DNA testing, it functions as a closed archive. Not from malice, from protection. The effect on the science is identical. Then, in 2023, a separate research team, completely unrelated to the Yates work, was analyzing pre-contact ancient DNA from southeastern burial sites. In the supplementary data, something appeared that did not fit the single migration model. They submitted it for peer review. It has been pending for 14 months. Not rejected, not published, pending. That word has a history in this field, and it is not a short one. Kennewick, Washington. Some physical anthropologist James Chatters submits a paper on Kennewick man's anomalous skeletal morphology to a peer-reviewed journal. It is accepted for publication. Before it goes to press, the Army Corps of Engineers seizes the remains under federal law. The paper is retracted. Chatters spends 9 years in litigation. He publishes his full findings 19 years after the discovery. Dr. Pontus Skoglund, then at Harvard, now at the Francis Crick Institute in London, published a 2015 study in Nature on a 12,000-year-old Brazilian skeleton known as Lucia. Her genome showed unexpected affinity with Australian Aboriginal and Melanesian populations, not Northeast Asian, a real peer-reviewed finding in Nature, cited 17 times in its first year. And funded follow-up studies on pre-Columbian Brazilian remains were not allocated by any major institution for 6 years after it published. The finding sat in the literature. Uncontested, unfollowed. This isn't a conspiracy. What it really is institutional gravity. Foundational models in science, especially models tied to national identity narratives, generate enormous inertia. You don't need to suppress a finding to make it disappear. You need only to not fund the follow-up, to not assign the graduate students. The drawer doesn't need a lock, it just needs to never be opened. The Cherokee Nation Tribal Council voted in 2010 to restrict access to Cherokee human remains held in university collections for ancient DNA testing. Their stated reason, cultural sovereignty, yet protection of their people's remains from institutions that have historically extracted and exploited them. This is not an unreasonable position. It is, in many ways, the only reasonable position for a nation that has been systematically stripped of everything else. The practical effect, the pre-contact Cherokee skeletal samples that could definitively test the Haplogroup T question remain unsequenced. The people most affected by the answer are the ones controlling whether the question gets asked. No villain, just an impossible geometry. Legitimate. That word does a lot of work in this story. It covers the distance between what is allowed and what is true. In 2021, a study in Nature Communications analyzed 161 ancient individuals from across the Americas. It confirmed the single founding population model for the vast majority of Native Americans. That's the headline. In the supplementary data, not the abstract, not the conclusion, Haplogroup T appears in 2 pre-contact samples from the southeastern United States, dated to approximately 3,200 BCE, noted as statistical outliers, not discussed in the paper, and yet the supplementary data said something the abstract refused to acknowledge. Dr. Theodore Scher at the University of Pennsylvania has spent years mapping Haplogroup X across the Americas. He confirmed the Siberian Gap. He has been careful not to claim transoceanic contact, but in every paper he has published on the subject, the gap remains unexplained, real, sitting in the literature like a question no one wants to formally ask. 3,200 BCE, the Chalcolithic period. Haplogroup T at peak frequency in the Levant, long-distance maritime trade active across the eastern Mediterranean. The question stops being whether a Levantine population could have reached North American shores. It becomes, what would it actually take to prove it didn't? The Appalachian Highlands, the Cherokee heartland, have been sampled at near-zero density for ancient genomics. The absence of ancient DNA evidence from that region is not evidence that Haplogroup T wasn't there before contact. It is evidence that no one has looked. Now, here is the part of this story nobody expected to find in a DNA video. Epigenetic research has shown that extreme population bottleneck events leave measurable chemical marks on the genomes of survivors, marks that are heritable, that can pass to children and grandchildren. The Trail of Tears, 4,000 people dead in a single winter, would generate exactly this kind of signature. Modern Cherokee DNA carries the biological scar of 1838 in its chemistry right now, in every living cell of every enrolled citizen. It also carries beneath that scar something much older, at the same molecular address. Cherokee poet Marilu Awiakta wrote in 1993, "The atom and the grandmother are the same. They are the memory the universe keeps of itself." She was writing poetry. She had never seen a haplogroup map. She was describing with complete precision what the science 20 years later would try to measure: address. That word is what the Cherokee Nation has been fighting over for 200 years. Not a location, a right. The right to define what their own origin means on their own terms. The Cherokee Nation is the largest tribal nation in the United States, approximately 450,000 enrolled citizens. It operates its own university, its own court system, its own healthcare network, its own language revitalization program. It has never commissioned an independent ancient DNA study of pre-contact Cherokee human remains, not one. The Eastern Band's 2017 statement on DNA testing is precise and correct. Commercial tests cannot determine Cherokee citizenship. The tribe does not recognize DNA as a basis for enrollment. Every word of that statement is justified. And for a researcher trying to understand where Haplogroup T came from before 1492, before the Trail of Tears, before colonization, before contact, that statement means the archive is closed, not from malice, from protection. The effect on the science is identical. Here is the geometry of the problem. The people most harmed by not knowing the full story of Cherokee genetic origins, people like the woman in Tahlequah whose test returned 0%, are Cherokee. And the people most positioned to open the question are the ones who have the most legitimate reasons to leave it closed. No villain, no conspiracy. An impossible geometry in which protection and erasure occupy the same space at the same time. Sequoyah invented the Cherokee syllabary in 1821, the only writing system in recorded history created by a single person, alone, without outside help, for an entire people. He built it so that nothing would be lost, so that everything, the stories, the ceremonies, the names, the memory, would survive. If you subscribe, what's coming next begins with a DNA result that a government tried to classify, a people who already knew the answer, and a question that science is only now catching up to. The Cherokee do not have an origin story that ends. They have an origin story that keeps arriving. in waves, in genomes, in the faces of people who take a test expecting one answer and receive instead a question so large it reorganizes everything they thought they knew. And the oldest part of that story, the part written before Egypt, before writing, before any of the names we use for ancient things, is still, right now, waiting for someone with the resources, the access, and the courage to ask it out loud.